It's common for blockages to develop in arteries that carry blood to the heart, which can lead to heart attacks and other serious heart diseases. About 600,000 stent procedures are performed each year in the U.S. to improve blood flow to the heart muscle. But there are issues with current stents. They can cause a blood clot, or the artery may become narrow again. This is why the National Institutes of Health is funding research at the University of Missouri, a total of $4.6 million for the development of a revolutionary stent. Engineers, biomedical scientists, and cardiologists are working together on this new stent. The research has reached the stage of animal testing. The stents are successfully implanted in pigs at Mizzou's College of Veterinary Medicine.
When we get into the heart and the coronary arteries, the arteries that supply the heart, in pigs the anatomy is identical to that in humans, both in anatomy and also in size. The response to having that stent implanted is similar in pigs and humans. So we can study the safety and how well that stent's working in the pig and get a very good idea of then how that's going to translate to what's going to happen in the human.
Stents are small, expandable metal mesh tubes that are inserted in blocked arteries to improve blood flow to the heart. There are two types of stents currently on the market. Co-investigator Dr. William Fay says the drug-eluting stent is covered with therapeutics to alleviate arterial re-narrowing. However, they can cause clotting with risky and potentially fatal consequences.
In the process of stopping the bad cells, so to speak, from growing that can make the artery re-narrow, they also slow the growth of the good cells, the cells that line the inner aspect of the blood vessel, vulnerable to blood clot formation. The other main type of stents are called bare metal stents. They're the same type of stents, but they're just not coated with anything.
With those, there are problems of re-narrowing and may need to be replaced over time. This new stent, called the Nanova stent, could avoid these issues. Dr. Fay says it has a special coating, helping to generate nitric oxide on the surface of the stent. The university team partnered with Nanova Biomaterials, a spin-off company founded by Mizzou researchers.
This unique process of coating the metal in a chamber with gases that deposit on the surface of the stent has been gradually developed by the Nanova team over about the last 10 years.
It requires using like a very low pressure, like almost like a near vacuum condition, but you still have some gaseous material. But the technique that we use is using like something called a plasma glow discharge.
John Eric Jones helped develop the company's plasma technique used for its nano-coating technology. In this test, he's simulating what happens in a catheterization lab, a technique used by cardiologists to implant the stent. Jones is responsible for the nano-coating molecularly binding to the stent.
We're using it to deposit like a carbon and silicon-rich coating onto the stents. You can further modify that coating to have like the proper bioactive agents. Some of the earlier modification techniques, the effect was very temporary, but I was able to find a way to sort of make the technique more permanent so that the modification, the chemical modification of the stent could remain in place, hopefully for the life of the patient once the stent has been implanted.
Blocking the re-narrowing of arteries and avoiding clotting problems. In a cath lab, the Nanova stent is implanted in a pig.
What we've learned so far in our studies is that the Nanova stent is as good as the state-of-the-art drug-eluting stent that's used in humans in preventing re-narrowing. But it also has an additional advantage that it appears as if it's going to be less likely to form a blood clot in the long term.
Over 100 pigs have the different types of stents, including the Nanova stent, for comparison studies.
The trials we've done so far give us signs that the Nanova stent may be less likely to cause those clots to form long term. We haven't done the long-term studies yet, and in reality, those studies most likely will have to come out of the human trials.
The results from healthy pigs are encouraging. The next step is studying how the stents perform in pigs with heart disease before human clinical trials, possibly in the next 3 years.
When we have to recruit patients and put the stents in them, this is where I can play a role in helping recruiting them, along with my colleague, Dr. Arun Kumar, who is the interventional cardiologist who would put in the stents.
This is a very exciting proposition for someone like me who's a basic scientist, because on this campus, we've basically gone from the materials phase in the engineering department in the lab to my basic science animal studies, and it's going to transition into humans. And we're going to be able to see that process all the way from the bench to the bedside, and to see the outcome of that sort of basic science work actually benefit the human patient. That's what we're here to do, and we rarely get to see that, and to have that potential is very exciting.
For Innovations, I'm Kathleen Berger.